Our method enables assessment of the composition and functional characteristics of different leukocyte subpopulations present in the uterine tissue of pregnant and non-pregnant animals.
This protocol allows for isolation of uterine lymphocytes while preserving surface expression of protein, cell viability, and functionality. Therefore, it is suitable for a range of subsequent applications.
Fetus removal at the beginning of the experiment and leukocyte collection are technically challenging steps. Since it is a lengthy experiment, particular attention and focus are needed once you reach the antibody staining steps.
To begin, transfer the uterus tissue harvested from a pregnant C57 Black 6 female mouse into a sterile petri dish. Then, using sterile instruments, gently remove the fat surrounding the tissue, taking care not to let the tissue dry. Remove the fetuses by dissecting each implantation site with sterile instruments, then return the uterus to its original 5 ml collection tube containing 1 ml of collection media. Using scissors, mince the tissue in the collection tube, then place the tube in a 37°C water bath. Next, add 3 ml of pre-warmed enzymatic digestion mix to the tube. Incubate the tube for 30 minutes at 37°C with agitation to enhance the enzymatic digestion activity. After the incubation is complete, vortex the tube and keep it on ice to inhibit the enzymatic activity. Then, transfer the digested tissue into a properly labeled 15 ml centrifuge tube. Rinse the